Thank you. Thank you, Linda, and thank you, Anjana and Vijay. I am going to talk about some theoretical work that we did to understand the spatial organization of eukaryotic chromatin. We heard a lot about bacterial chromatin, and I'm going to talk a little bit detail about how they are organized. So, what we know certainly is that in eukaryotes in particular, the spatial organization of the chromatin encodes information that decides the fate of the cell. What do I mean by the fate of the cell? For example, as the cell differentiate, what will they end up as? Will they end up as, for example, a skin cell or a brain cell or different types of cells? All of these cells have exactly the same DNA, but these cells behave very differently. And the reason why they behave differently is basically because of the chromosome organization and the gene regulation resulting from the chromosome organization. So the assembly of chromatin in these cells decides the fate of the cell, while the genetic code is exactly the same. So the important question is how this chromatin is organized in space, as well as they are dynamic, so they change in time. So how do they change in time? And the three-dimensional organization matters. They are relevant for function. For example, they are highly relevant for gene regulation, events like recombination, translocation, repair, and spreading of histone modifications, etc., etc. There are many other functions that is dependent on how chromatin is organized in 3D. As a theorist, to understand the 3D organization, when we say 3D organization, it essentially means some kind of structure, but it is not like a crystal protein structure here, which is thought to be kind of static, but is highly dynamic, but it does have on an average certain structure. So what one can compute is a spatial distance, accurate spatial distance between different segments of the chromatin. So can we predict from simulations and theoretical studies the 3D spatial distance between any two segments of this long chromosome in mammalian cells in particular? And that is relevant for many functions, as we said. And one of the experimental methods to do this, and one of the popular methods that works very well, is something called the chromatin conformation capture experiment. And roughly the way it works in the following way. So you have this long chromatin, you add some cross-linking agent, which is kind of shown as red color here in this cartoon. And once you have this cross-linking agent that cross-links every region that come in proximity, you can cut using some cutting enzymes the different parts of the DNA. For example, think of this cross-linking, and you can cut at four different locations, and you can separate out this X-like segment and do some processing, which I wouldn't go into. And then you can sequence this X-like part. You can see that this portion will come at one part of the genome. So you compare with the existing human genome sequence. So you could do the sequencing. This would come somewhere, and why this part would come somewhere else. So you can ask how often you got two segments X and Y in proximity, which are at some location on the genome, and one can make a matrix. So essentially at the end of this experiment, one gets a frequency matrix or a contact frequency matrix, which says what is the probability that there was a contact between two segments X and Y, which is somewhere along the genome far away. And it's a symmetric matrix because probability of contact between X and Y is same as Y and X. And such experiments, which is now a high throughput version of this called the HiC, is a population average experiment where you average millions of cells and the data is generated from this, right? And the question that as a theorist one would want to study is that given this kind of two-dimensional matrix, how do you go back to the three-dimensional shape of the chromosome? And it's a little bit non-trivial because of the following fact that these contact probabilities are not one. They are not always in contact. They are contact for a very short time, or they are contact with a very small probability. So these contact probabilities are very small, and given this that these contact probabilities are very small and the system is dynamic, it is not easy to go from this picture to this picture. So once you have this, you also want to answer this question that what is the 3D distance between any two segments of my choice, any two genes or any two segments. And there certainly polymer physics matters. So now if you want to understand this, one has to understand chromatin as a polymer. and one has to have a polymer description of chromatin. We have a reasonably good polymer description of DNA, and something called the worm-like chain model became quite popular in the last 20 years or so to describe the DNA. And why is it popular? Because we understand all the parameters of the worm-like chain model, like the persistence length, the twist persistence length, and the intrinsic curvature, and so on and so forth. But none of this is known for chromatin. We do not know the polymer parameters of the chromatin at all. And we cannot quantitatively describe chromatin without really understanding what the parameters of these polymers are. What we know from the textbook is that chromatin is a string of nucleosomes, and that's kind of well established now, right? So, and one has a nucleosome level description of chromatin, at least as a string of nucleosomes. However, what we can't do is that we cannot simulate the entire chromosome as at the nucleosome scale, because there are millions and millions of nucleosomes. Like one chromosome itself is more than 1 million nucleosomes, and the whole genome is like 20-30 million nucleosomes. So we cannot study the whole chromosome or whole genome at the nucleosome scale. So one has to have a kind of a coarse-grained model to study chromatin as a polymer. So the question is, how do we bring or make a polymer picture of chromatin? One has to take a few nucleosomes as a bead and connect them with springs and make a polymer picture to simulate and study chromatin. How do we do that? To do that, we don't even know basic things like the size of a bead. If I just take a bead, let's say a few nucleosomes as a bead, is it 25 nanometer? Is it 35 nanometer? What is the size of this bead is not at all known. And depending on how the chromatin is locally folded, the size of the bead could be very different. We don't know what is the stretchability of this chromatin. In other words, what is the spring constant? We don't know. We don't know what is the interaction between different segments of the chromatin. Are they attractive? How strong these attractions are? So basically, we do not know much about the physical properties of the chromatin, and we would like to understand this from some data. So since we know something about nucleosomes, and when we were thinking about this problem, this micro-C experiment data came up. Micro-C is the high C at nucleosome resolution, where one can get the contact between any two nucleosomes or at 200 base pair segments. So we started with this micro-C data from mouse embryonic stem cells. And so this is the contact map from the micro-C. Here, one dot represents contact between two nucleosomes. For example, this dot represents a nucleosome at 31.22 MB and 31.24 MB. And once you have such a contact map from experiments, can we generate a polymer configuration from this? There are a couple of different methods which we have we studied in the lab, and Sangram adopted one of those methods, which is a simple method, which is take this contact map, follow some procedure. In that procedure, essentially the procedure ends up with the following task. You have to insert contact between any 2 points, but not always, but with the contact probability. So for example, if this dot has a contact probability which is shown here of, let's say, 0.1, then you will put a spring between them only in 10% of the realizations. So you have an ensemble of configurations. In only a fraction of the configurations, you will have that spring. So, but there are some extra details which I'll tell you in person after this talk if you're interested in, which is essentially to take into account the fact that there are some correlations in this whole problem, and we thought about all that and we put in those correlations in some particular way which I can describe. But the idea is that insert such interaction springs, for example, with a contact probability Pij, and then equilibrate the system using Brownian dynamic simulations. And once we do that, we can get a bunch of configurations whose average would look like the contact map, right? So if you do simulations and if we find the contact map from the simulations, this contact map would look exactly like the contact map from the experiment. What we essentially did, basically because we put this in by hand, right? So we have a bunch of configuration whose average would reproduce the contact map. Now, one can ask the question, once we have these configurations where each bead is a nucleosome or a 200 base pair region, what is the distance between any two regions? So you can take a region here and you can take a region here and ask, what is the average distance between them? Because you have a large number of configurations, there will be distribution and an average. And Adhya did this for in detail for many regions. For example, you can compare this data with microscopy data for certain regions. And it turns out that you can compare it would compare very well from our simulations. And the correlation, they are like essentially correlated very well. So one axis is the microscopy data, one axis is simulation data, and there's a one-to-one comparison. And our prediction is highly correlated, very well correlated with the experimental data that you can measure with certain microscopy methods. Once we have that, we can then ask if we have this detailed configurations. For example, for function, what matters is the location of, for certain gene regulation function, what matters is how often certain regions like enhancers and promoters come together. So here what is shown in this green is a gene region, and what is shown in the red is an enhancer region. And these two are two different configurations of the same polymer, of the same gene segment, Col1A1, in HFF cells. And in these two cells, these enhancers and promoters have different configurations. So in here, they are kind of clustered, while here the enhancer is quite extended. And these are just two example configurations. We have large number of configurations, and we can measure the distance between enhancers and promoters. And we can ask the question, which configuration is relevant for function? And we are discussing this and doing this in collaboration with Dimple from NCBS to understand how to make sense of gene expression and connect it with chromatin organization in these cells. Now, once we have this nucleosome resolution configuration, we can do a process called coarse graining, which is basically considering a segment, like which is, for example, here 10 nucleosomes as a big bead. So you can take a small polymer, which is in the red color here, which is a part of the genome. You take this 10 nucleosome and draw a sphere around it. And you ask the question, what is the size of this sphere, the radius of gyration? How big is the sphere? And you can draw spheres along the genome. Every 10 nucleosomes, you can draw spheres. And you can ask the question, what is the distance between center to center of these two neighboring spheres? And this center to center distance, which is called the LCG here, and its fluctuation. Since we have an ensemble of configuration, you will get a distribution of this LCG, and this distribution would tell us what is the spring constant, how flexible these segments are. And so we have the size of the segments, let's say 5 kb or 10 kb, and you have the distance between them and their distributions, which tells you how flexible these regions are. And then you can look at 3 such coarse grain beads and look at the angle. And then you look at the distribution of the angle. And this fluctuation in the distribution of the angle will tell you how bendable these regions are. So we can get some idea about stretching elasticity and the bendability of chromosomes, starting with micro-C data, which is the nucleosome resolution data, and constructing a polymer which is consistent with the micro-C data. And studying an ensemble of configuration that we constructed. So we did exactly this. So we have, for example, the size of 1 kb bead along the genome. So x-axis is the genome location and y-axis is the size. So you can see nanometers, 28 nanometer, 27 nanometer. On the right-hand side, you can see it in nanometers. So if you take a 5 nucleosome, which is 1 kb, this is the size of the genome, of the segment. If you take, for example, 100 nucleosomes, which is 20 kb, this is the size of the region, and so on and so forth. So we can get for a different segment of chromatin, how big would that polymer or the bead would be. And we can plot the average of this as a function of the size. For example, what is shown here is the genomic size, 10 kb, 20 kb up to 100 kb. And this is how the radius of gyration of the size would look as a function of the size of the segment for different genes. So different color represent different genes, and the bluish color represents heterochromatin, and the reddish color broadly represents euchromatic genes. So this gives us some idea that how big a particular segment of the chromosome chromatin is. And this has an interesting correspondence with the contact probability map. For example, at the boundary of these domains, these sizes are very large, suggesting that at the boundary, the chromatin is extended, while inside the tags, the chromatin is compact, and so on and so forth. And this has implications in many other things that we computed. Now, one can ask, as I said, what is the spring constant or what is the stretching elasticity of these chromosome segments? So we can look at the distribution of the distances between two neighboring coarse grain beads and compute the stretching elasticity. And the spring constant can be plotted as a function of the coarse graining genomic size or the coarse graining size. And we have it in piconewton nanometer per micrometer here, or is some dimensionless unit here. And we can also predict how does the spring constant change depending on what scale of chromatin you study, whether you are studying chromatin at 1 kb or whether you are studying chromatin at 20 kb. You might need to use different spring constant because the flexibility locally would change, and we can get some sense of it. And one other interesting point is that if you take two segments of chromatin, for example, if you take a 5 kb chromatin, which is marked in red color, and another neighboring 5 kb in blue color, they would mix. There are proteins that making them contact, plus the entropy would make them mix. And this would imply that in polymer simulations, one shouldn't put a Lennard-Jones-like interaction which would not allow them to mix. Rather, one should put some kind of a soft interaction which would allow them to mix. And the overlap between this is estimated through some method here. And I'll be happy to tell more about this method soon, later. We can compute other structural quantities like dihedral angles, like the Ramanujan angles in protein. We can compute equivalently for chromatin, and we have done that. And we have distributions of bending angles and dihedral angles, and these angles are very wide. And this wide distribution suggests that they are quite flexible at the scale of several kb. Now we can ask the question, what is the potential of interaction between two segments? And one can use some kind of a method called an iterative Boltzmann inversion method to obtain the interaction potential between two different segments of chromatin. And we did that, and we found that the interaction is a soft interaction, which is much softer than the Lennard-Jones potential. So what is black here is the Lennard-Jones potential, and this is the interaction potential that we got through this iterative Boltzmann method, and this is quite soft. So once we put in all these parameters, spring constant, interaction potentials, and so on and so forth, one can predict the 3D distance of the size and of a bigger region accurately. And that's what we went on to do. So Adhya predicted the organization of the entire chromosome at chromosome 21 level. And this is the Hi-C map of the chromosome 21. And we reconstructed the entire chromosome in a coarse grain model at that level, and the map looks the same. And one get very good map of the 3D distance as well, which I haven't shown here. Then one can assemble such chromosome into multiple chromosome, which I wouldn't go into the details. We can ask the question, what is the interaction strength of these two segments? And this interaction strength is related to the contact probability. So we could come up with a empirical relation between the contact probability Pij and the interaction strength epsilon ij. So given the contact probability, we can compute the strength of the interaction, how strong that interaction is. And this makes the simulation very easy, relatively easy to start with, because we have some idea about interaction strength given the contact probability. And then one can get many interesting relations, like what is the 3D distance as a function of contact probability? If I give the contact probability, can I say something about the 3D distance? They have a relation which appears like a power law. Of course, it has a wide variability. It's not a neat power law as one would expect, but one can draw an overall power law there, and it has some resemblance with a power of kind of something between 3 and 4. The 3D distance goes like p power minus 1 by 3 or 1 by 4, or more like 1 by 3.5. So we do get this relation between contact probability and the 3D distance. Now, given this structural information, one can ask the question about the dynamics. How do these domains form? How do these domains get organized? And there are two models. One is the loop extrusion model, which says that there is this particular motor, which is a SMC complex, which binds and extrudes the loop and generates this 3D structure. And there is also formation of condensates, as just like Ranjith talked, and many other proteins are known to form condensate. And these condensates can also organize chromatin, presumably. So there are two models. And today I'll briefly talk about this loop extrusion model. So the aim of the simulation that we did is to predict the time-dependent changes in chromatin as a function of loop extrusion parameters. So loop extrusion has some set of detailed processes like binding of this loop extruding factor with some rate. So I would go to the next slide. So it has several parameters. Binding of the loop extrusion factor here cohesin with some rate R plus, and dissociation of this factor with some rate R minus, and they can extrude with some rate R sliding, RS, and they stop at the CTCF boundaries, which is kind of a non-phenomena in this area. So given these parameters, we can vary them in simulation and ask the question, how does this parameter affect this 3D organization? So we have studied them in detail. So for example, when for some parameter regime, the TAD would not form properly. Only for certain parameters, you will have the complete TAD formation. And then one can ask the question, how do different proteins, so these are proteins known in this field called VAPL and NIBBL, they increase either the loading of cohesin or they promote dissociation of cohesin. So we can introduce effect of these proteins through these parameters, these rates. So we vary these rates, which is equivalent of varying these protein concentrations or expression of these proteins, and we can ask what would be the effect of this protein and predict them. Then one can ask, what is the relaxation time, which is a property of a polymer, which is one way of getting some time scale from such polymer dynamics. So one can compute the relaxation time as a function of genomic separation. From a simple model, one would think that the relaxation time goes like the length square or something close to the L power 2. But for chromatin, depending on the extrusion parameters, one can get a series of, one can get different values starting from S power 0.9 to S power 1.7. How does the relaxation time would scale as a function of the genomic separation between two regions that where they form loop, right? And one can then ask, even with extrusion, what is the role? You have extrusion, but what is the role of intrachromatin interactions? So intrachromatin interaction does have a role. So intrachromatin interaction is needed to get appropriate TADs. And then one can ask, what is the mean square displacement? So this, what is plotted here is the mean square displacement of two loci, which basically says how much area do they explore, right? So one can get, how does the slope of this tell something about the dynamics? So the mean square displacement for different conditions, for example, wild type, when you mutate cohesin, which means you stop extrusion, when you don't have the boundaries, which is the delta CTCF, when you have some loading, unloading, different rates, how does this MSD, the root mean square displacement, vary? And we can compare these experiments, some of those single molecule live microscopy experiments, and it turns out that our predictions are in agreement with many of some of these experiments. And one can then study other aspects of how extrusion compete with intrachromatin interaction in a crude model. And we studied that competition between extrusion and intrachromatin interactions. So you can generate such contact maps without extrusion and with extrusion. The contact map would look the same, but our claim is that with and without extrusion, even though the contact map looks the same, the dynamics could be very different. So even though the contact map is the same, the dynamics could be different with and without extrusion. And this is the claim for some regions where extrusion and intrachromatin interactions compete. And one can, in the last 1 minute, I'll just say a couple of examples where we can connect 3D organization to dynamics and function. Vinod, who's here, studied how this 3D shape would affect spreading of histone modifications, and you can talk to Vinod. Anirudh, some student in the lab, recently studied how does this 3D organization would affect translocation frequency, and it would change some relation, the prediction for the translocation probability, Tij would change, and we have a detailed relations of this, and I'll be happy to tell about it, and double strand break dynamics and few other aspects we can study. So to summarize, we talked about what are the polymer properties of chromatin, the bead spring chain model that described chromatin, and we can predict bead size and stretching and bending elasticity, loop, and we studied loop extrusion dynamics, and we can connect 3D organization to dynamics and function, 3D organization dynamics to function. And I'll be happy to take questions and explain more this after the talk. These are the students who did the work and the collaborators and the funding agencies. Thank you.
Thank you very much. Questions, Srikant?
Ranjith, great talk. I have lots and lots of questions, but I'll ask one. In this method of iterative Boltzmann inversion,
yeah,
I guess the assumption is that you have a thermal system, right? And in the context that you're looking at this, there is a lot of, I guess, understanding about sort of driven behavior and so on. So is there any sort of systematics about how you would actually do an inference of effective interactions in that situation?
Yeah, so here what we assume that given a set of interactions, one would simulate some equilibrium simulations and one would get this structure. That's the assumption. But how do you do this in a purely non-equilibrium case? I don't know. Maybe we can talk about it. I have no idea how to do that in the non-equilibrium. Here, this is that you have some interchromatin interaction with some strength and whatever, and you put in, you would get the equilibrium, the structure, the chromatin structure as an equilibrium property. That's the assumption.
No, I mean, you could do the sort of the converse, which is to do the experiments with dead cells. Right?
Yeah.
That's another possibility. Anyway, we should talk.
Yeah.
Can the next speaker go set up their talk while we're continuing the discussion? Yes, Anjana, please.
This was really interesting. I have a question about how much information on dynamics one can extract from your analysis. So I'm wondering whether you're getting information on dynamics within a given cell, or you're inferring the various states of various cells within the population, and that then gives you some understanding of dynamics within the cell.
So the dynamics that we did here, the simulation can be thought of as the equivalent of a single cell. What we would, so for example, what we did, we have a polymer in which we do the extrusion, and the parameters of the extrusion are kind of probably the average of the cell. I don't know whether different cells will have different parameters. So that is kind of an average of the cells, but given a set of parameters of extrusion, that means how fast they will extrude and how fast cohesion, what is the rate of binding and dissociation of these complexes that extrude, then we can get equivalent of a single cell.
So the reason I'm asking this is that the output in the comparison is to a population averaged high C.
No, not for dynamics. Dynamics is for live. For dynamics, we compare with live microscopy single cell experiments. Yeah. But for the high C, it's a population. We also do for the population. We generate large number of configurations and we average.
But following up on that, so you mentioned that in between the TADs are the region of highest variability.
Yeah.
But the TADs are a function of the average. And do they really exist in single cells is a major question of interest to many of us. So how does that change your interpretation of the regions of variability if the TADs are just an illusion of averaging?
Yeah, so this is a good question. And of course, all of these questions, we don't know the answer. But given the CTCF boundaries and the binding of CTCF, there is some indication that TADs might be there in the single cell as well. For example, there was Javed Zhuang's lab, they did the microscopy, right, on single cell, like the DNA paint. experiments, and they do see TAD-like structures in the single cell as well. There is some indication that probably they exist, but with some probabilities, not that all the time they will be there. With some probability, there will be TADs in single cells. Yeah.
Any other questions? Okay, well, thank you again for a very interesting talk.